you cannot, it's because I cannot see you guys actually here if I'll be doing like this. I cannot see messages or chats. So will it be okay if the screen is as big as this one? Okay, maybe just like this. Klaro pa rin pag ganito kalaki?
Yes po.
Okay.
Yes, sir.
Yes po.
Very good. Now let's go here and let's start here. So basically, we're going to talk first about the functions of your cells, and you already know this one. You've encountered, you've learned this one during your high school days, that the cells are the basic unit of our life. Okay, and every living organism actually has its cell. Okay, now let's talk first about the function. First is the synthesis of molecules. Now, synthesizing of molecules, I think we've introduced this one already in your chapter 1 if you already watched it. Okay, this cell synthesis actually various types of molecules, and then including your proteins, your nucleic acid, and your lipids. And the different cells of the body do not all produce the same molecule. Okay, remember in our first discussion, we've talked about atoms, right? And then the atoms, when they do chemical bond, they can actually form your molecules. Now, these molecules, if they also fuse or bond with other molecules, they can actually synthesize or they can make your cells. Okay, so that's the function of this one. Next one is communication. Okay, if you're going to relate this one in our previous topic in chapter 1 about the organization, like everything in our body is interrelated and they are communicating from one another. And the primary enabler of this one is actually our cells. Okay, because our cells actually produce and they receive chemical and electrical signals that able to communicate from one body part to another. And also we have your reproduction and your inheritance. Okay, so it's because we have in our genetic information as individuals, we can actually transmit this one with our offsprings, with our kids. Okay, everything is okay so far?
Yes, sir.
Masyado ba tayong mabilis or sakto lang yung pacing ng lecture?
Medyo mabilis, sir.
Okay, bagalan natin ng konti, ano? Okay lang mag-Taglish or English or Tagalog?
Tagalog, sir, para mas maintindihan po.
Sir, Taglish.
Taglish na lang. Mahirapan ako pag mag-Tagalog tayo purely. Okay?
Okay, sir.
Sure.
Awesome. Very good. Now let's go here. First, we're going to talk actually the cell structure. Okay. What is the composition or the structure of our cells? And again, we're going to start with your organelles. Okay. Remember in chapter 1 that the molecules, when they bond together, they will form organelles, right? Itong organelles, ito yung tinatawag natin ng mga tiny organs. Okay? And this one, they actually have your cells. And this one, they specialize structures in cells that perform specific functions. We have the example of nucleus, your mitochondria, and ribosomes. Actually, we will be discussing these examples later on, one by one in details. Now let's go to your next one, which is your cytoplasm. This one is the jelly-like substance that holds your organelles. So actually, it is being encapsulated in your cell, encapsulating your organelles. Okay, I will be showing you an illustration of this one here. Nakikita niyo itong picture na ito. So this one is this far end here. This is your cell membrane. Yung cell membrane, tinatawag din natin yan na plasma membrane. Okay? And this is the outermost part of the cell. And next to that is itong cytoplasm. Ito yung diniscuss natin kanina na jelly-like formation. And this one is actually protecting our nucleus. Okay? Clear?
Clear po.
Nakikita ba natin yung arrow kapag ginagalaw kong ganito? Yung
Yes
Yes,
po.
cursor?
sir. Yes,
Okay,
sir.
that's better. Thanks. So let's go here. So let's talk about your cell membrane. So don't be confused if narinig nyo yung cell membrane. It is also the same as your plasma membrane. And it is the outermost component of the cell. Kumbaga sa isang buong human being, ito yung ating skin. Ito yung outermost part ng ating katawan. Sa cell, ito ay tinatawag natin na cell membrane. And what is the function of this one is your selective barrier. And it encloses your cytoplasm. Selective barrier, it means that siniselect na lang niya yung pwedeng pumasok. Kung meron kang quarantine pass or wala, hindi ka makakapasok. So he is selecting who can enter and go out inside your cell. And it says here, it encloses your cytoplasm. Let's go back to our image. This is the cell membrane. Next to that one, the yellowish part here, that is your cytoplasm. So outermost cell membrane, next to that is cytoplasm. Okay. Now it says here we have your extracellular and then intracellular. Now, again, what I want you to remember in learning medical terms, you go to the root word or base form of the word. Like here, extra means outside, intra means inside. So whenever you hear extracellular, it means outside the cell. When you hear intracellular, it is inside inside the cell. Claro?
Yes po.
Yes po.
Okay,
Yes po, sir.
Yes, sir.
very good. Now let's go here. Let's talk about now the structure of your cell membrane. So it is called your fluid mosaic model. I know you heard this one already in your biology, siguro in your senior high school or grade school. Yung fluid mosaic model, it's because it looks like water. That is how the cell membrane looks like. And it actually made out of your phospholipids and protein. Now, later on, doon sa illustration natin, ipapakita ko sa inyo yung pagkakaiba at saka yung mukha talaga nitong ating fluid mosaic model. It says also here that phospholipids form double layer or bilateral. And the phospholipid contains two regions. We have your polar and also your non-polar. Okay, let's discuss that one here. So if we say polar region, it is the head part of the cell membrane, okay? And it is hydrophilic. Now, remember, if we say hydro, it means water, and philic means loving. So what is happening here is this is exposed to water, okay? Because it loves water. It is hydrophilic. Now, the nonpolar is the opposite of this one. It is the tails, okay? And it is hydrophobic. If we say hydrophobic, phobic means he fears the water. And this one is away from water. Okay, this is the illustration of this one. As you can see, this is the head. The head is polar and it is hydrophilic. This is the tail. The tail is nonpolar and it is hydrophobic. Now let's talk the composition. Itong head usually contains phosphate, okay? While that tails contains fatty acids. Okay, I hope this one is clear. Now let's go here. So this one is the one I'm talking a while ago. It forms two layers or bilayer. Layer one here, nauna yung head and tails, and then here, tails and then the head there. Okay? Na-imagine na how it looks like. Ito yung ating fluid mosaic model. Is this one clear? Okay. And as you can see here on this part, this is the head part. It means yung head natin is polar. So this one is exposed with water and it likes water all the time. This ones, yung tail naman natin, they hate water. Okay, so that's how are you going to distinguish your polar and non-polar. Let's move here. Okay, so you can actually see this one, this table on your book, but we will be discussing it one by one here. So let's skip this one first. And let's go here. Let's talk about the movement through your cell membrane. A cell membrane selectively determines what can pass in and pass out of the cell. A while ago, I already mentioned you about this one. Now, the enzymes, glycogen and potassium are found higher concentration inside the cell. So whenever they will ask you, okay, how's the concentration inside the cell? What are the compositions that you can see there? You can say they are the enzymes, glycogen, potassium. How about outside? You can say your sodium, calcium, and chloride. And that's how the movement of a movement through your cell membrane. Here, ways or molecules pass through the cell membrane. Paano naman nakakapunta or nakakadaan yung mga different things inside or outside of our cell? So first here we have your what we call your directly through or the use of your diffusion. Are you still familiar with diffusion? Anyone? Do you still know what is diffusion? Or nakalimutan na natin? Or hindi nyo pa na-encounter ito?
Sir,
Yes,
is to yung movement ng individual, sir? Yun ba yung ano po?
yung diffusion.
Fusion po, sir. Movement of
Yeah.
individual?
It's actually ito yung movement ng ating oh yes, someone raised a hand. Sino yung nag-raise ng hand natin? Yes, Miko.
So yung diffusion po is generally composed of two major parts, the solutes and the solvent.
Very good. So yung diffusion is actually is being used in your it's because remember in our laboratory before in our senior high school or in our grade school, we always do your we always have your solution, right? And yung solution meron siyang two major parts, yung solute and your solvent. Yung solute yun yung how do you call this one? Ito mm, yung
Yung ions.
dinidissolve. Yung dinidissolve. While your solvent, ito yung nagdi-dissolve. Okay? And what is happening there, yung process ng movement doon is what we call your diffusion. So what you always remember here is from an area of higher concentration to area of lower concentration. Example here are your oxygen and your carbon dioxide. Okay. Next, we have your membrane channels. Para sa naman itong membrane channels? Protein that extend from one side of cell membrane to another. Side, shape, and charges. It can be positive or negative ions determine what can go through. Look at this example. The sodium can actually pass on the sodium channel. It's because they have the same composition. So they can just easily go through that channel. Okay, and we call that one as your membrane channel. Let's go to the next. Okay, as you can see here in this illustration. Okay, so this is a membrane channel. A while ago, kanina sinabi natin yung sodium, it can easily go through to the sodium channel. Like this one. So this is a specific non-lipid soluble. It will just go directly here. It can just pass through this one. It's because they have the same composition. While this one, the non-lipid soluble, it will bounce back. It's because they don't have the same composition. Now let's go here. Carrier molecules. Ano naman itong carrier molecules natin? They bind to molecules, transport them across and drop them off. So it's like riding a bus or riding a taxi going to a specific place. Okay, it's because they will just drop you off after you ride in that vehicle. That is just like your carrier molecules. Example here is your sugar or your glucose. Another example on how to go in and out of the cell membrane or your cell is through the use of your vesicle. Okay, vesicle is like a sac-like structure and can actually transport a variety of materials. What is happening here in vesicle, it's because they can just easily fuse with the cell membrane. Okay, I think we have illustration here. Not this one. Okay, this one. So as you can see, this one is the secretory vesicle formed at your Golgi apparatus that is actually moving toward the membrane. So papunta siya doon. But this one, what is happening is, look at the second picture. The secretory vesicle membrane fuses with the cell membrane. So you can see here, it is actually starting to fuse. Okay, and once they completely fuse, the materials here can easily be released outside our cell membrane. Is it clear how does your vesicle is being utilized? Klaro tayo doon?
Yes, sir.
Okay, very good. Now let's move here. Oh, we have actually here the definition of diffusion. Again, it is the movement of molecules from areas of high to low concentration. And a while ago, Miko already mentioned about your solution, solute, and solvent. Solution is mixture, example ng solid, liquid, or gas. Solute is ito yung ating dinidissolve, while solvent is ito yung ating pang-dissolve. Like your water or vinegar, those are kind of your solvent. So example here, we're going to add salt to water. Now water will be your solvent and solute will be your salt, and the mixture, we call it as your solution. As easy as that. Okay, so this is an illustration here. The salt here, this is your solute, and the water is actually your solvent. Okay, as you can see, what is happening is there is a movement of higher concentration of solute to a lower concentration. Now let's go here. How about your concentrated gradient? Measures concentration difference at 2 points. These things are just good to know. Filtration is movement of fluid through a partition with holes. We will learn more about this one in the next part. Okay, how about this one? Your mediated transport mechanism. Narinig nyo na ba itong term na ito? Mediated transport mechanism.
Hindi pa po.
Okay.
Hindi pa po, sir.
Now, what is with this one? What is your mediated transport? This one is diffusion with the aid of a carrier molecule. That is the facilitated diffusion. It means that we don't need energy for this one. It requires no ATP. Are you familiar with ATP? Have you heard ATP before? Yes, anyone?
Opo, sir.
What is ATP?
Adenosine triphosphate po.
Okay, very good. So that is actually your adenosine triphosphate. In facilitated diffusion, we don't need ATP. While in your active transport, we actually require the use of your adenosine triphosphate, especially during the sodium-potassium pump. Okay, we're going to discuss that one here. Okay, look at this illustration here. This one is, can you see it clearly?
Yes po.
Okay, so this one, the carrier molecule binds with a molecule. Like this one is the carrier molecule, and yung molecule na gustong pumasok, example is yung sugar, they will just bind here so that it can pass through your this one plasma membrane. Okay. The next one here is the carrier molecule actually changes the shape so that it can accommodate the molecule that is passing through. That's how are we going to use your carrier molecule. And here, mm, this is an example of your active transport. Again, with the active transport, what are we going to use? We need actually your adenosine triphosphate, right? So what is happening here, if you're going to look here, this is your sodium potassium pump. So dito, meron tayong sodium pump. Tapos, kailangan niyang lumabas. Paano siya makakalabas? So, kakailangan natin actually ng adenosine triphosphate. Now, once adenosine triphosphate will release an atom, it will become diphosphate. Kasi nga nirelease niya ng isa, so nabawasan. So magiging diphosphate. And this one will actually aid in releasing your sodium. Okay? And eventually, after releasing that one, and also your potassium over here, and that's how are they going to transport one another with the use of your adenosine triphosphate, while here we don't need the use of your ATP. Okay, just like this one. This is another illustration on how to use your carriers. Okay, next is here. We're going to talk now about your osmosis. I know you already heard this one also. So osmosis is the diffusion of water across your cell membrane. And here we actually use we have your term osmotic pressure. This is the force that required to prevent the movement of water across your cell membrane. And as you can see here, this instrument here, this beaker actually who has water and has what is this? Is this salt or what is happening here is the water is going up. Okay, that's how the osmosis is being utilized here. And we have the types of osmotic solution. Okay, so this one, you need to learn this one as early as today. It's because you will use this one in your higher years, especially if you are on duty. Like you will be giving intravenous fluid to your patient, and you should identify if that fluid is hypotonic, hypertonic, or isotonic. So let's learn the difference between among these three. Okay. So first is your hypotonic. From the word itself, hypo, it means mababa. Okay, so in hypotonic, the lower concentration of solute outside the cell, and it has higher concentration of water outside the cell as well. Okay, so what is happening here is the water is moving out from the cell. Okay, now, I mean, the water here moves in the cell. Pumapasok yung tubig. Imagine that if you are drinking a lot of water, and then eventually the cell will swell. Okay? Kumbaga mamamaga. In hypotonic, it's because there's always a constant introduction of water in the cell. The cell is actually swelling. Okay? And this swelling will actually lead to lysis. It will burst. Kumbaga, sasabog siya kasi punong-puno na siya ng water. That is your hypotonic solution. Well, here, your hypertonic, hyper means mataas, concentration of solute outside the cell, and the higher concentration of water inside the cell. What is happening here is the water is moving out. Okay? If the water is moving out in our body or in the cell, what is happening is we are being dehydrated, right? So bumababa yung tubig natin sa katawan, nadi-dehydrate tayo. So what will happen there is it will actually shrink. The cell will shrink. Liliit siya, which is what we term as your crenation. Okay? Did you guys follow the difference between hypotonic and hypertonic? Is this one clear?
Yes, sir.
Sir, pwede pong paulit.
Yes, sir.
Yes, sir.
Okay. Sige, ulitin natin. Pag hypotonic, hypo means mababa. It means yung tubig pumapasok sa cell. Pag pumasok yung tubig sa cell, nag-swell, namamaga. Eventually, pag sobra yung swelling, naglalysis. That term is yung burst, sumasabog. That is for hypotonic. While hypertonic, mataas yung concentration, yung tubig lumalabas sa cell. Pag constant na lumalabas yung tubig sa cell, ang nangyayari, nadidehydrate, nag-shrink, lumiliit yung cell, and that is what we call your crenation. Is it clear now?
Yes, sir. Thank you po.
Okay, awesome. Now, we still have another one, which is your isotonic. So isotonic, this one has equal concentration. What is happening? The water doesn't move and the cell is remaining intact. Okay? So kumbaga ito yung sakto lang, isotonic. Now we have illustration here so that you can easily understand the difference. So example, this is actually your RBC, the red blood cell. And what is happening here if we put RBC in a hypotonic solution? As you can see here, it is because it is hypotonic, the cell is actually swelling. So there's a tendency that it will,
Burst po.
yes, it will burst. Very good. Now, let's go to the far end. This one is your hypertonic. So the RBC being introduced to hypertonic solution, the water keeps on going out to our cell, so it will shrink. It will go smaller. While here in the middle, your isotonic, it's like there's some sort of balance. Okay, are we clear now with the difference of hypotonic, isotonic, and hypertonic solution?
Yes, sir.
Yes, sir.
Awesome, very good. Now let's go here. Let's talk now about your endocytosis. Now, what are your endocytosis? Endo means inside. Okay, so endocytosis, this is the process that brings material inside the cell or into the cell using vesicle. Remember, we've talked already about vesicle a while ago. So we have two types of your endocytosis. The first one is your phagocytosis. Okay. The next one is your phagocytosis. The difference here, in phagocytosis, they're cell eating, especially the solid parts. So they are engulfing the solid particles. They're eating it. While your phagocytosis, they are drinking it. Okay, they're drinking the liquid parts. So that's just the difference between these two. Let's move here. How about your exocytosis? Exo means outside, palabas naman ito. the process that carries materials out of cell also using your vesicle. Okay, example is this one. So if you're going to look closely with this illustration here, this is an example of your exocytosis, yung palabas ng ating cell. And what is happening here, if you go look closely here, again, they will fuse, the vesicle will always fuse with your cell membrane so that they can go outside. Okay, so that is what is happening here. This is an example of exocytosis. So this one, vesicle, the vesicle is carrying particles going out. So go out, then eventually here, they're fusing on this part until on this third one, they can go outside the cell membrane. Okay, and this term, we call it your exocytosis, going outside. Okay. Okay, just like this one. How about this? This is your receptor mediated endocytosis. Okay, it means that we are using a vehicle like this one, receptor molecules here, and then eventually they will go here, and then they're already all inside. Okay, so let's go back here. So we've finished your cell membrane and your cytoplasm already. And this time we're going to talk the cytoplasm. Oh, I think we've finished this one. Cytoplasm is this part here. So again, it is inside the cell and it is like jelly-like fluid. And what is the function of this one is give cell shape. And again, it will hold the organelles in place. Now, we also have your nucleus, this one, your nucleus. What is inside your nucleus and what is the function of this one? It is actually houses our DNA, okay? And it is the center of the cell, the nucleus. Okay, look at this one. Look closely at this illustration. So this one here, this is your nucleus. And we have here your nuclear envelope, this part. So that is encapsulating actually or protecting our cells. So this one is the nuclear envelope here. And we have this one is the interior. And you can see the nucleolus here. And this is your chromatin. We're going to discuss this too later on. Okay, what is the nuclear envelope? It is actually on the edge of your nucleus and the nuclear pores. It is in the surface of the nucleus where materials pass in and out of nucleus. So it's like cell membrane of nucleus, your nuclear pores. Okay, as you can see here, nuclear pores is this one. On the edge part of the nucleus. And this is the nuclear envelope that we've discussed a while ago. Now, how about this one? Let's go to your chromosomes. Chromosome, the location of this one is inside the nucleus, and it is made of DNA and protein. And the function of this one is the part of our genetic makeup. While your chromatin is also inside your nucleus, and if it's loosely coiled chromosome. Look at this one. Look at this illustration here. Kung nakikita nyo ito, so yung chromosome tsaka chromatin, di ba, nasa inside siya ng ating nucleus. And this one, your DNA, is actually coiled. Nakabuhol-buhol siya. And it is compact. And if this one looks like this, we can actually call that as your chromosome. Pero pag tatanggalin natin yung buhol-buhol, yung coil, we can call that as your chromatin. Okay? And as you can see here, this is our DNA. Now, what is your nucleolus? Your nucleolus is also inside your nucleus and it produces your ribosome. Okay, now what is ribosome also? It is attached in your RER. So whenever you see your RER, it means your rough endoplasmic reticulum or your cytoplasm, and it produces your protein. So during exam, what produces your ribosome? It is your nucleus. What produces your protein? It is your ribosome. Okay. How about here? Okay. This one, you can see this illustration on your book. Now let's go here. What are the difference between your RER and your SER? Your RER rough, S is smooth. Your RER has your ribosome, while your SER, it doesn't have ribosome. That is why it is smooth, because they don't have ribosome. Both of this one is actually in your cytoplasm. Okay. And again, the characteristic RER, it has ribosome attachments, while SER, it doesn't have ribosome. While your RER is the site of our protein synthesis. In SER, site of your lipid synthesis. Okay, I hope this one is clear. These are the difference. As you can see here, the dots here are your ribosomes, and the folded part here who has dots are actually your rough endoplasmic reticulum. And here on this part, they look smooth. They don't have your ribosomes. And these are your smooth endoplasmic reticulum. Clear?
Yes, sir.
Nasusundan pa natin?
Yes, sir.
Medyo nakakalito, sir.
Which part? Para ulitin natin. Yung difference ba?
Yung difference po, sir.
Okay. So basta pag rough endoplasmic, remember, meron siyang ribosome. Here, yung dot-dot. Pag smooth, wala siyang ribosome. Yung RER, site ng protein synthesis. SER, site ng lipid synthesis. Okay? Now let's move here. Okay, ano naman itong next? This is your Golgi apparatus. Your Golgi apparatus is on your cytoplasm. So what is the role of this one? The main role of your Golgi apparatus is closely they are packing stocks of the characteristic is closely packed stocks of membranes. And their function is nagkukolek, nagsusort, nagpapackage at nagdidistribute ng protein and lipids. They're like the Lazada, or they are the ano na yung isa? Shopee. Yan, yan yung ating Golgi apparatus. Sila yung nagko-collect, nagsusort, nagpa-package, and then nagdi-distribute ng proteins and lipids that eventually our body is utilizing. Okay, remember that one, your Golgi apparatus. And we also have your secretory vesicle. Location is your cytoplasm. This one, these are the one naman na nagdi-distribute ng materials out of cell. Okay? Sila yung how do you call this one? Distributor. Okay? Sila yung JNT, or other package dealers. Next one here. Okay, as you can see this part, these are the folded things that the characteristic of your Golgi apparatus. And here, it actually produces your secretory vesicle that can move out materials outside our cell. Okay, and we also have your lysosome. Lysosome, these are the location is cytoplasm and the enzymes that digest foreign materials. So it means they're like eating the foreign materials in our body. Okay, example for this one is maybe there is a bacteria. And then if there is a bacteria, the lysosome is actually acting out and ini-engulf niya yung bacteria. Kinakain niya kasi nga foreigner yung bacteria. Okay? Next one is we have your mitochondria. This is in your cytoplasm. It contains our folds or the cristae, and it produces your adenosine triphosphate. And where do we use again your adenosine triphosphate? During your active transport. Okay? And this one, your ATP, actually has a lot of use. You know, it is the main energy source in many chemical reactions that is happening in the cells or in general in our body. Okay, so when they ask you in the exam or in the quiz, who produces your ATP, it is your mitochondria. Okay, as you can see, this one, this is formation of your, this is how lysosome work. Example is this one. This is a foreign body that went inside of us, and then eventually the Golgi apparatus will produce your lysosome. Then the lysosome will go here. It will go this foreign material, and eventually it will engulf it and it will digest this foreign material. And this one, this is the cristae in your mitochondria. Okay. Are you familiar with cilia and flagella? Narinig niyo na ba tong terms na ito?
Not yet, sir.
Not yet, sir.
Okay. So maybe you will encounter this one also in your biochemistry. Okay, if we say cilia, it is location is in the surface of the cell, and this one, it looks like our eyelashes. Okay, cilia looks like our eyelashes, and the function of this is to move the materials across the cell surface. While your flagella, this is a cell surface. This one is meron siyang whip. It looks like a whip. Ang function naman nito is minumove niya yung cell. Example is yung sperm. Di ba yung sperm, kung nakita niyo na yung how does the sperm looks like? It has whip, right? Para siyang may tail. So that is actually, the flagella is actually moving or helping to move your sperm. And we also have your microvilli. This one is shorter than your cilia. Okay, it looks like shaggy hair. And it increase surface area. So these are your the things that we actually use to transport other materials in and out of our cells. So your cilia, flagella, microvilli. Cilia, it looks like our eyelashes. Flagella, it has your whip, while microvilli is smaller than your cilia. Now let's move here. Cytoskeleton. Remember, if we say cyto, it means cell. And if we say skeleton, it means framework. Okay, so from the word itself, this is the cell's framework, and it is made out of your proteins. And the functions of this are support, hold the organelles in place, and enable the cell to change your shape. Cytoskeleton. So we have three types of your cytoskeleton. We have your microtubules, intermediate filaments, and microfilaments. In order for you to appreciate this one, so this part here, this is your microtubules. It looks like a tube. And here is your intermediate filament. And the last one here, that is your microfilaments. Okay. So this things here that you can see, they actually give shape to our cells. Okay. So example here of your intermediate filament actually is the keratin. If familiar kayo sa keratin, yung protein na associated sa ating skin or sa ating hair, that is an example of that one. Okay, so as you can see here, this is an example of a microtubule triplet. See, there are three microtubules here. So these are the things that giving shape to our cells. Okay, how about your cell activity? What is happening there is the cell's characteristics are determined by the type of proteins produced, and proteins function is determined by our genetics. And the information in DNA provides the cell with a code for its cellular processes. Most likely, this one, you already encountered most of this, especially if you are in your strand is stem. You actually learn this things already, and we all know our DNA. Okay, DNA is double helix in nucleus. It composed of your nucleotides, and also it contains our 5-carbon base or 5-carbon sugar. And this is how it looks like. Okay, this is our DNA. So the strands here of the DNA separate each other, one DNA strand, and serves as the template for your mRNA synthesis. Most likely you've learned this one already. Now, how is the flow of this one? Flow of genetic information, we call that as your central dogma. Okay, it occurs in 3 stages. We have your DNA replication, transcription, and your translation. Okay, here in DNA replication, how it is being replicated from, you can see this one, this part here, this is the original DNA strand that we have. And on this process here, it's actually being separated. The DNA strand here is being separated. And as you can see here, we have a new nucleotide. And here, this is the old strand, and then the old strand. And then here, we are forming new strand. Okay? With that, these things will fuse together. And when they fuse together, we have new DNA molecule here, and then the new molecule on this side. So that's how DNA replication is happening. Now, the gene expression, this contains the information in DNA, directs protein synthesis, and a protein provide code for gene expression, and enzymes regulate chemical reaction and uses transcription and translation. Let's look at this one. So kung nakikita nyo ito yung DNA natin. And then here on this part, magi-start na yung ating transcription. Kung nakikita nyo dyan sa ating board, dyan magi-start yung ating transcription. One DNA strand result in your mRNA, which is complementary copy of your DNA here. And then on the third part, and then here, yung number 4 naman, yung amino acid natin, building blocks of protein are carried to the ribosome. Pupunta naman siya sa ribosome, and then in the process of translation, the information contained in your mRNA is used to determine the number of kinds, arrangement, and the amino acid chain. Okay, yung transcription, ito yung the process which DNA is read. And it usually occurs in your ribosomes. And it produces your mRNA. The M in RNA here is your messenger. And it has 3 sets of nucleotide base that code for a particular amino acid, and we call that as your codons. Here, just like this one, while your translation, ito naman yung process ng messenger RNA is kinu-convert into amino acid, okay, and produces your proteins. So codon pair with anticodons. So yung anticodons nito is tatlo din. That is carried by your tRNA. Your transcription, it is carried by your mRNA or produces mRNA. Like that. Okay, cell division. Are you familiar with this one? Have you learned cell division before? Hello? Anyone?
Opo.
Okay, what is cell division? If you've
Parang,
learned this part. Okay, go.
parang cell replication lang din po.
It's simple as that, right? So parang nadi-divide lang siya. So this one is actually yung paano magfo-form ng two daughter cell from a single parent. Okay? And this one, I know you already learned your mitosis and your meiosis before. Each cell except your sperm and the egg contains 46 chromosomes, or what we call your diploid. And sperm and eggs contain 23 chromosomes. Now, mitosis, ito yung cell division that occurs in all except sex cell. And it forms your two daughter cells. And we have here three terms that you need to know: your chromatid, centromere, and your centrioles. Your chromatid, two strands of chromosomes that are generally identical. Your centromere, where two chromatids are connected, and centriole are small organelles composed of 9 triplets. Okay, just like this one. Okay, how about mitosis? Familiar na kayo sa interphase, prophase, anaphase, tas telophase? You've learned this one already, right?
Yes, sir.
Okay, so this is just like a quick review of what you have learned before in biology about the stages of your mitosis. So in interphase, what is happening here is DNA strand, the chromatin, the replication actually occurs. And it is next is your prophase and the metaphase, anaphase, and telophase. And this is how it looks like. Okay, like at the end of the process, you will be, or there will be these things here. You can actually see this one on your book, and it's easy to understand your cell division. Okay, so are you guys clear with this one? Okay, so that's actually or basically the end